in the space of visual system development and function. He started as an undergrad all the way through to a PhD student at Cambridge, telling me earlier it was in zoology, so that's sort of fits. He then did his first postdoc with Leon Legnano before going on to the lab of Thomas Euler in Germany, before moving to Sussex in 2016. So Tom has, again, made a lot of seminal discoveries in the field of visual system development. It's probably easier for me to list the things he doesn't work on when it comes to color vision than it does to name them all. But essentially, he's been one of the pioneers in using zebrafish to study high acuity vision, for example, cone adaptability, prey capture, so on and so forth. And he's going to tell us a little bit about his recent work on color vision today. Tom has won a number of awards, the Eppendorf Prize, the Lister Prize, amongst a number of others. So he's very well decorated in that space. But also, I think what should be mentioned is Tom is a big proponent of open science. And he was a very early adapter of bioRxiv, for example. Everything goes on there. You've started open software platforms. And you basically share everything you do, including probably this seminar soon online, right? So I think that's a really commendable thing for science nowadays, is really sharing everything. And Tom's at the forefront of that. The other thing that Tom does, which I think is particularly impressive, is he started Trend for Africa, or Trend in Africa, which is a nonprofit organization where he basically science training and communication in Africa. I think there's over 40 academics who contribute to this. It involves training in the field, but also acquiring equipment that will allow them to, start to work towards cutting edge science education in Africa. And that's something that Tom started several years ago. So that's also a very impressive feat, I think, and something that Tom should be certainly recognized for. So with no further ado, I will turn the seminar over to Tom, and we'll hear all about his really amazing work. So I will start this, and I will give you a pointer. It's all yours.
Alrighty. Well, thank you very much for the invitation and the introduction. I won't be talking about Africa or open science, and I will barely talk about color vision because having worked on color vision for so many years, I've kind of gone off it because I think color vision is an epiphenomenon. Let me explain what I mean by that. Okay, clicking doesn't work. This does. So this might as well be on the cover of a textbook on color vision, right? So that's kind of how we think of it. So we've got our rods. There's one type. Doesn't really matter what spectral wavelengths they're sensitive to because there's only one, which means you can't do spectral comparisons. You can't tell intensity independent from wavelengths. That's the definition of color vision in most people's books. And therefore, everything that we see at night is gray. And you can test that, go to the forest at night, it's gray. It's just correct. However, when you switch on the light or you wait till the daytime, then we get to use our cones, which are less sensitive than the rods. The cones, as it so happens in the human eye, come in 3 spectral flavors. That means that if we were to take the signals from these cones and compare them rather than add them together, we can make a spectral comparison that is different from just an intensity measurement. And based on that, we can see color, or we can tell wavelengths that depend from intensity, and our brains interpret that as color. So that's great. So based on this, what you might conclude then is therefore the presence of multiple cone types enables color vision. And I think that's true. If you have multiple cones, you can do this. That's fine. But I think what we've been doing, and I think it puzzles me how this hasn't been challenged before. It kind of, what we always say that therefore the purpose of having more than one cone type is to have color vision. But that makes no sense. Because they could have evolved for something else, and then we just use them for color vision, right? So we've kind of flipped our causalities, and it doesn't get challenged. And what I'm hopefully trying to convince you of today is that at least the original purpose of having multiple cone types is to enable something as fundamental as vision, so aspects of vision that are not about color. And they are specifically important in the water where vision first evolved, right? So all eyes almost all eyes across animals have evolved in the water first, and one day they came out of the water in a species such as ourselves. And then you just work with what you inherit from your aquatic ancestors. And I think that's also the case for our vision. So if we take our little diagram here of our rods and cones, rod and cones, and we take our combination contrast, we feed them everything into the vision box, and we say, this is vision. And then once or multiple times, probably in the history of evolution of vertical vision, this system has then become co-opted to also allow color vision. But this is like an optional branch. You don't need it. Humans use it, but some species might not. But quite fundamentally, this just happens because the spectral information that comes in through these different channels is not identical. It's actually quite overlapping, but it's not identical. Therefore, if for any reason other than color vision, you take two of these cones and you contrast the signals, you're going to end up inevitably with a spectrally complicated signal. Some sort of opponency will happen. And if it's here, used for something useful, you could also use it then to, in addition, get a sense of color vision out of it. So I think this is sort of a more plausible history of color vision. And now I need to convince you of that. So here's water and the rainbow. And hopefully what you can see is that this rainbow is very rainbowy in the top and not very rainbowy in the bottom, right? There's nothing magic about this. This is just physics. This is the interaction of light with water, depending on the wavelengths, i.e. energy of the photon that you have. You interact with water stronger or weaker. There are some wavelengths that penetrate water better than others. That means that, well, color or the wavelengths composition is informative about all kinds of stuff in the water. And this would have been the evolutionary pressures driving everything to do with how we set up our detectors in vision. Now, what we've done here is a very simple thought experiment. So here, faint in the background, what you see is the average spectral content of light in shallow, fresh water. So it's not muddy, it's just like basically where zebrafish live. It's like 0.5 meter deep, it's clear water, it's lovely. Stick a spectrometer in there and you just see what's the wavelength composition. Yeah, and hopefully it's quite, and notice that flipped here. So normally what we plot is we plot red over here and UV over here. But here, this time I've got red here and UV over here because this is an energy axis, which is inverted wavelength, just to make my argument here. And what I've plotted on top of it is the in vivo spectral sensitivity functions of two of the photoreceptors of the zebrafish, the ancestral red cone and the ancestral UV cone. And hopefully, and we've measured these using two photon with all kinds of tricks in that paper. So hopefully what you can appreciate is the red cone is basically a copy of all the light, right? The red cone is a light detector. It just sees the light that's there. The UV cone is doing something completely different. The UV cone is very specifically trying to get the bits that is actually UV. Okay, so you don't really have a red and a UV system. You have a gray and a UV system, if anything. Now, starting on that premise, we might be able to then just plot these two cones in this arbitrary space of how many photons do you have. The red gets a lot more photon than UV just because there's a lot more light when you measure all the light. But the UV cone gets the good photons, the high energy photons. UV is the highest energy light we can see. If you go higher energy, that didn't, well, it doesn't get through skin, it doesn't get through anything, right? So there's a sort of biological limit of how far you can push it this direction. So this is the best of the light, but this is the most of the light. And you can use those for different purposes. So for example, you could say, if I can count a lot of photons, that means that I can shorten my integration time. Therefore, my photoreceptor becomes fast. Therefore, if I want to do fast vision, you really want a system that catches all the light, like the red cone. The UV cone is terrible at speed. But what the UV cone is really good at is gain. The reason it's good at gain is because the energy is so high that you don't run into thermal noise, right? So this is one of the key things. There's a reason that we don't see infrared. Infrared is really low energy light. And it's so low that the energy coming in to try to photoconvert your opsin, your retinal, is so low that it gets matched and swamped by the thermal energy of the system, right? So if we operate something like 38 degrees in your eye or whatever, you just can't push the opsin further into an infrared region. You will just start seeing nothing. You will run into thermal noise. That problem exists on this end. It doesn't exist on this end. On this end, the problem is cancer, right? So as long as you can keep your DNA damage at bay, you want to go that way. And that means that a UV cone really hasn't got much thermal noise at all. Right? You could use it as a photon counter, it would be fairly clean. And that means, though, that even though there's not a lot of photons in the UV, you can take your gain and you crank it up all the way as much as you want. You're not going to run into noise. Okay, so you've got a completely different detector system. You've got a fast, low gain system, and you've got a slow, high gain system. And this is not the properties of the photoreceptor yet. This is the properties of, this is just what inevitably happens if you build a detector that works in a certain wavelength range. And it's not just invertebrates, it's true pretty much across any species you look that has multiple spectral receptors, they're always like that. Now, what then becomes quite interesting, I think in vertebrates at least, is that the vertebrate eye, of course, doesn't have the photoreceptors in isolation, it has them in the circuit, and the circuit does things. And the first thing that the circuit does is it takes these photoreceptor signals, which are already different, and it pulls them apart and makes them more different. So for example, here the photoreceptors in the zebrafish eye, as in most vertebrate eyes, are contacted by horizontal cells. Horizontal cells do interesting things. If you take your horizontal cell, if you record a red cone, this is a red cone glutamate response to the stimulus. You can see it follows the low frequencies and eventually gives up. And then you block the horizontal cells, nothing else. Then you clip the high frequency response, right? So what that means is that the presence of the horizontal cell, through whatever mechanism, I don't want to get into that, makes the cone faster, right? That's the purpose of this system. If you do the similar experiment for the UV cone, we didn't use a chirpy, we just used a flash of light. It goes the other way around. The cone is normally quite slow, is slow in the presence of horizontal cells, and when you block them, it gets quicker. Okay, so the horizontal cells make the red cones faster and the UV cones slower, right? They take the intrinsic differences and make them bigger, suggesting that this is a difference that you want to work with. Okay, so we've moved the dots a bit. Then let's talk about space. So if you have a textbook and you look at how a cone is supposed to respond to light in space, it's supposed to have centers around organization. You hit the outer segment of the cone, it responds negatively because these are off cells, so they decrease activity. But if you hit it near the center, but not on the center, it should go actually the other way around because there's an opponent circuit, there's an inhibitory circuit that goes the other way around. And in red cones, we can reproduce this, right? So here's a very simple experiment. You've got little red bars, we're recording red cones, and it keeps missing. And here it hits it, and then misses again, right? So this is just the central receptive field. It's quite small, it's a couple of degrees, makes sense in the baby zebrafish because the eye is so small. But then what you can do is you can contrast invert that stimulus and play the same game. And you will find a place where the black bar sits exactly on the cone and the red bar sits exactly not on the cone. That causes the, that hits the opponency, and then you flip the signal. You turn it into an on cell, you know? So this is textbook stuff. This is what should happen with a cone. It's a center surround unit. What center surround does is it makes, when it decorrelates in space, it makes your ability to encode edges in space better. Yeah, and the entire visual system is kind of based on that principle. It's just it starts in the cones. You try that in a UV cone, it just doesn't work, right? So here, this experiment is equivalent, except that we're recording from a different photoreceptor. And hopefully you can appreciate that the receptive field is huge, it's something like 10 degrees. This is crazy big for a photoreceptor. Bear in mind that the outer segment of the photoreceptor is not 10 degrees, it's maybe 1 degree. Okay, so something's going on there. I think it's chromatic aberration. So basically you can't focus all wavelengths the same. And the eye just lives with that. And then that becomes basically a massive detector. But also we just can't, oops, I haven't got this here. We can't get a surround out of this. So we've tried this experiment, doesn't work at all. We've tried all kinds of clever other experiments, doesn't work. We've never managed to invert the polarity of a UV cone based on a cleverly designed spatial stimulus, implying that either there is no surround, or it's so weak that we can't detect it. Okay, so what that really implies then is that the way
there's a tiny little thing, the center and the surround of the red one,
yeah,
they don't seem to be in the same place or near.
It's a different cone. Sorry, yeah,
oh, it's a different one. Okay, go.
Yeah. No, yeah. Good point though. Yeah, so they just do completely different things. They're in a different position in speed, in a different position in gain, and they're in a different position in space. They're basically two completely different ways of sampling the world. Yeah. And if you sort of mentally translate into what that would do, the red is basically your detail channel, right? It sees everything as well as it possibly can be seen, pretty much. Like if you take a grayscale camera and you just point it under the water and you have enough pixels, that's roughly what it'll tell you, plus some processing. You get a nice black and white image. UV image by itself will also give you a black and white image, right? There's no color computation. It's just, it looks at the UV bit, but it'll have a really high gain. It'll be quite slow, which also drives the gain up again. And it'll have terrible spatial resolution, right? It's just, it's trying to look at a completely different thing. It's a very sensitive channel, but it's not a detailed channel. Okay, so it seems then that this is sort of the principle of feature selectivity in the visual system, right? Like the idea usually is that you have where the photoreceptors detect the light, and then it goes to the bipolar cells, the ganglion cells, goes into the brain, does all kinds of things, and then you get these features. And these features might be, you've got a big receptive field, you've got a small receptive field, you've got one that is very sensitive, one that's not very sensitive. Well, it looks like the codes are kind of doing that for free, right? Like the visual system starts with these features, but this is just two of them. Okay, so you might call both of them by itself. They're brightness channels, they're black and white channels with different properties. Now, what about the other two channels? Zebrafish, like many surface-dwelling teleo, or basically anything that swims in the water that evolutionarily never left the water, has four cones. That's the ancestral state. There's the green and the blue. And a while ago, we did similar experiments for the spectral sensitivity function that I showed you in the beginning for these, and it turns out that these are color opponents at the first synapse of vision, right? So basically, these guys, through the horizontal cell network, they don't tell you greenness or blueness. They're just a color opponent signal. They respond positively at one wavelength and negatively in another wavelength. Now, if you've got a color opponent system, the mind immediately goes to color vision, right? We know that the currency of color vision is color opponency, and that's true in the humans as well. So that's how we wrote up the paper and published it and thought that was great. But actually, coming to think of it, I think that was short-sighted. I mean, yes, it will give you color vision if you choose to use it that way, but it's so much more useful for much more basic things. So if you think about this gradient again, the gradient doesn't just work down, it also works along the water column, right? If you stick your head in the water and look along the coral reef, you're going to see this. You see color where you are, and you see blueness where you're not, right? And it works for the fish as well. That fish is near, that fish is far, okay? So the spectral composition of an underwater image tells you about distance. Now, I would posit to an animal that swims around the water, unless it's a very specific purpose, distance is more important than color, right? Because distance allows you to interact with the world long before you understood what you're interacting with. It stops you from running into things. It stops predator getting you, stuff like that. So, working on that assumption, let's think about what these wavelength channels would do with color. Well, first of all, red sees all the light. Therefore, it sees the furthest, right? There's just no way around that. UV actually doesn't travel very well in water at all. Therefore, if you only have a UV channel, you don't see very far. So if you only have a red and a UV channel, you will get two distance shells just for free. But then these color opponent signals, they will actually travel intermediate distances. And because the color opponent system don't have an identical zero crossing, there are two different zero crossings, it will give you two different distance shells. So kind of just for free, by the way that these channels are set up, you will get four distance shells. If they're spaced like this, I don't know, but something like this. And it would be crazy for fish to not have evolved to use this. It seems like the cheapest way of doing distance, which incidentally should work without even having any decent spatial resolution. Like this, you could do with the single pixel detector that just measures different wavelengths. You could do distance estimation like that. So if we then think conceptually as the input channels in the zebrafish, not just as four color channels, but basically as two completely different two different grayscale channels on the extremes, plus a couple of different distance channels here, I think it makes more sense. And I just want to point out, so what I've done here, this is a GoPro image, so it's red, green, blue, human optimized, right? You stick it in the zebrafish natural habitat. This is actually taken in India. And this is what it looks like when you look at when you just pool the channels. This is what it looks like when you take a red channel and your green channel and you flip the green channel and put them together, right? I didn't do any magic here. I just made a color opponent, a fake human-centric color opponent signal. And already you can see the foreground and the background just disappears, right? So these color opponent computations, they're really powerful tools to get distance information. They're basically foreground enhancers. Now, working on that, so if this is our basic model, it's not very satisfying though, is it? It's just a bunch of boxes. So how can we unpack this and really understand what's under the hood? So my proposal is this. We have in the retina of fish, but also in humans still, two main systems. We've got the ancestral red system, which goes together with rods. You can ask me about why I think it goes with rods later. Just take my word for it for now. And on the other hand, you've got the UV system, which goes by itself. And they give you the high resolution versus high sensitivity systems. And they can individually drive certain behaviors. So for example, optomotor, optokinetic response type things are generally associated with long wavelength vision. And I think it's actually just the red cone circuit. Prey capture in zebrafish, for example, is a UV circuit, right? So there are examples where really the cone seems to be the key thing. And I would argue that this is all of vision. Why would I say that? Just the detour, just to make the point that ancestral red cones in zebrafish are both red and green cones in humans, and the blue cones in humans is the ancestral UV system, right? So humans don't have the green and the blue, the ancestral ones. They've got the ancestral red and UV ones. And this is important because it's true for pretty much all vertebrates. So here's a little phylogenetic tree of most major branches of the vertebrates. Up here, you've got squidgy little fish, more interesting looking fish. Then we start to get into lizards, amphibians, and whatnot. We've got the mammals down here. And hopefully what you can appreciate is that if you look at the rod, always there. Right? There are reports of rodless species, like a chameleon, was thought to be rodless. Turns out it's wrong. They just don't have very many. So they've been missed. So I'm not aware of any vertebrate that doesn't have rods. Not even hagfish, which barely have an eye. They still have rods. Then red cones is the next best one. Almost all vertebrates have red cones. The only ones where it's unclear is lanternfish, live very, very, very deep. Coelacanth, very, very weird and very distantly related to anything that's alive today. And hagfish, barely an eye. Okay, so basically red cones are always there. And red cones will forever shift their spectral sensitivity to match whatever makes sense. So for example, in the human case, there's the red and the green version, but it doesn't stop them from being ancestral red cones. They're just different versions of it. And that happened actually a bunch of times in the evolutionary history of animals. Okay, so the red cone is almost always there. Next best one is the UV one. It's like the red cone, except you've got some special cases like seals, whales, basically large aquatic, secondarily aquatic species that have lost it. And again, these weirdies. And the sharks and rays, which are a little bit, well, they're basically the size of a dolphin, similar problem. So almost all vertebrates have this system. And that is entirely untrue for this system. The blue and green cones are missing in pretty much half of the lineages. None of the mammals have them. There's a report of platypus. I think this is probably wrong. And you can ask me in a year or two after we've checked it. If the platypus is wrong, all of the mammals are done. There's no survival of this system. And there might be a new occurrence in the standard, which is a marsupial. Very unclear. Basically, mammals don't have this system.
Can you clarify what system we don't have with mammals? Because I mean, we do have green and blue cones. So can you just clarify?
So that's what I meant here. That our green and blue, what we call to be the green and what we call the green cone is an ancestral red cone with a green shifted opposite, and what we call the blue cone is an ancestral UV cone with a blue shifted opposite. Like, for example, marmosets, lemurs, they will have this, but also cats, dogs, horses, basically frogs, crocodiles. It happens quite often that this UV opposite shifts into the blue range, but the photoreceptor itself is the ancestral UV one. Yeah. So, and hopefully, if I organize them a little bit from live in the water, live sometimes in the water, don't live so much in the water. And hopefully you see there's a bit of a pattern there. It's like it's a water system. It survives in some animals that don't live in the water, like some birds, some lizards. I'll get to that if I have time. But basically, it's an ancestral water system. And what I would posit is that this is not used in how we think cones are used. I'm saying that this is used as a regulatory system. We know in the retina we've got horizontal cells as lateral connections. We've got amacrines as lateral connection. I posit that these middle cones are part of that system. They're the regulatory system. They're not the drive system. And I know this is heretical, so I will justify it. So how do you test this? So let's go back to the distance. So there's a lot of aspects that you could test, but we thought the distance one's maybe the sexiest to look at. So how do we think about distance? Well, actually, the color disappears with distance. That's quite clear. But color is the epiphenomenon of a much more simple phenomenon in physics, and that is the narrowing of the wavelength with distance, right? Stuff that's near, you have a lot of wavelengths available to make spectral judgments. That's why things do look colorful, but really it's the whiteness that disappears. In the front, stuff can be white, but if you take a white t-shirt and you start swimming into the distance, it'll be a blue t-shirt, right? So the spectral width is the easiest to measure factor that changes. So just to illustrate how that it's really powerful, right? So here's an RGB movie. These are artifacts. These are not real, right? So ignore that. So basically, you've got a little underwater movie, again, where the Tabak live. What we can do is we can just extract the brightness component by just adding all the channels. And yeah, us being clever humans, we can tell where's the foreground and where's the background, right? But it's cognitive. You need to think this through, right? If you just look at the whiteness, so if you just look at how broad is the spectrum based on these 3 pixel, like the RGB values that are available in the video, I think it's really obvious that the foreground here is super high whiteness, and it just very obviously decreases. And I like to point at this particular hulm of grass or seaweed or whatever it is, right? It sort of comes out of the nothing. So the whiteness really is a very powerful and easy way to signify distance or lack of distance. So how would you measure this? So if you are a fish, you could use different photoreceptors to pick it up. So let's start with the actual spectrum. So here's just the hypothetical spectrum. It's not what it actually looks like. It's just the so if this is super near you, as you go further away, the spectrum will shift towards a much more narrow one, typically biased towards the long wavelengths. Right? So this is near, this is far. This is roughly what will happen. If you have a spectrally broad non-opponent system, like the one down here, it'll just copy the spectrum more or less, right? So you lose excitation. There's less light to integrate, therefore your signal goes down, but it goes down a bit, right? As you make this narrower, you just lose a little bit of signal. But the opponent systems, they're more interesting because in the beginning, when you've got a broad spectral input, the negative and the positive will cancel and you get nothing, right? So opponent systems are silent under white light. But as you get rid of some of the wavelengths, but not others, that's when the opponent systems kick in, right? So the opponent systems actually start to be more and more active up to a point, and they go down again, right? So if you have an opponent system in your eye and a non-opponent system in your eye, and you just contrast them, right? You basically say, I'm going to take my non-opponent system to drive everything, and I'm going to use my opponent system to regulate the system. Then all of the entire output of the visual system will become white biased. Like stuff that you do will work really well if it's white, and stuff that you do, if it's not white, your responses are going to go down. Your retinal output is going to go down, right? So you don't need to do clever cognitive computations or anything. You just need to take an opponent and a non-opponent system, contrast them, and make that the output of the eye. Yes.
Is there an assumption here that the reflectance of the objects that you see is going to be very broad? For example, if all objects were blue, this would not work.
You're completely right. Yes.
Okay, so what is your assumption there? Because I wonder whether actually you're assuming that it's flat, that there are objects of all colors. I'm using the words that we would use for color in the sea, in the ocean, in the I don't know. This is fresh water.
This is fresh water. In the ocean, yeah.
So yeah, you're assuming that.
Yes, but actually, if you walk around nature with spectrometer and you point it at things, almost everything gives you some sort of broad spectrum with a little bit of a kink this way or this way, right? So if you point it at a leaf, it goes this way. If you point it at the sky, it goes this way, but it never goes like this, right? Whereas if you point it at a children's toy or something, perfect red, then it goes like this, right? So it's man-made objects that are like this, and the occasional occurrence where animals or plants have evolved to try to be narrow, like some plants, for example, some feathers of birds, they are narrow. But they come much later in evolution than any of this would have been important. And actually, there's very little evidence for the existence of structural color on pigmentation of skin or anything in old things, right? So the first clear evidence for the existence of, shall we call it purposeful color, is something like 300 million years old. But the eye would have evolved 550, right? So it's quite a difference. But you're completely right. Yes, it doesn't work. If everything is monochromatic, then this fails. Okay, so how do you test this? So we have tested this, and I'm going to give you the punchline before I show you. First of all, using zebrafish, we see that zebrafish vision, is white biased, and it's not a little bit white biased, it's crazy white biased. If you give a zebrafish white light, everything works better. 2, I'm going to show you that the red and the UV system is necessary and sufficient to drive all of vision. So when you take it out, vision goes away. As long as it's there, vision is fine. The green and blue system, completely different. You can take out the green and blue system, vision is fine. And actually, some aspects of vision improve. Or the gain goes up. Improves maybe the wrong word. So the green-blue system is not necessary nor sufficient for vision. And it's actually pegged in a mutually competitive way to inhibit vision. 4, I'm going to show you what that means in behavior. And 5, if you've got time, I'm going to talk a little bit about evolution. So here is finally your standard zebrafish experiment. So here's a zebrafish. That's the brain. Lots of neurons. They're all expressing GCAMP. And we have a 2-photon microscope to record the activity. We scan this bit of the brain. This is the tectum. This is the other tectum. In fish, all the information goes across, right? So if you stimulate them from here, all the activity will be here, specifically in this red zone, which would be the retinotopically aligned bit of the brain from where our stimulus is. Then we've got a custom stimulator that can do, it can give it fish white, so real white, or all kinds of combinations thereof. Here's the actual stimulus that we use. So here is the spectrum of light in nature. That's the average one. And here's how we've placed our spectral channels in power and wavelength. And hopefully you can see we're trying to approximate this white light. Right? So the idea is that if we pop on all 4 spectral channels at the same time, we're approximating the natural spectrum. I'm going to call this white. If you do the exact same thing, but you take out the UV, I'm going to call it visible. So this is what we would use maybe. But this, for example, would be a UV-only stimulus. And the experiment now is very simple. Having set this up, we're just playing a stimulus like this, right? We're showing it some gratings that start to move. Then we're switching off the light. We're showing some dots. This would be like a prey capture type stimulus. And then we're doing a contrast inverted thing of the same thing on with the dark dots. And we're just seeing what can we get. Okay, so this is now in white. And here there's a bunch of example cells. So for example, this is a cell that responds to gratings. Every time you show gratings, it responds. It doesn't respond to anything else. It's a grating cell, simple. This is a dot cell, responds to light dots. There's non-cell, there's not cell. You find all kinds of things. Most cells are quite selective for one of them. Sometimes you get 2, but that's actually an edge case. Most are pretty selective. Now, where in the brain are they? Okay. The grating cells are in the retinotopically aligned area of the brain, where they're supposed to be. The dot cells are also in that part of the brain. The on and off cells are all over the place. So it looks like they, either they don't come from the eye, all of them, because the fish has other photosensitive bits, or if they come from the eye, they get distributed in such a way that they reach the whole brain. Which one of the 2 it is, I don't know. But hopefully, just the location of the cells shows you very clearly that this is not the same system. It's something else.
Or is the full retinotopic map within the area?
Yeah, so the screen is roughly where the, yeah. Okay, but I took away my punchline, but here's my punchline. We take the white, we delete the UV and leave everything else intact. So we're just taking away this tiny little bit of light here. And you see how this breaks, right? The grating in your dot response, yes, there are still cells, but the proportion of cells compared to what you get with white is pitiful, yeah? Whereas this on and off system isn't, I mean, it's affected a bit, but not so much, okay?
So when people study zebrafish vision, do they usually use a UV monitor?
Some do, it's rare.
Okay, but if they don't, they're missing a lot.
It is a criticism that would be valid, yeah.
Yeah.
You said it, not me. Okay, so, but then you might say, well, maybe UV is amazing. Maybe UV is the remaining 85% here, and then we solved the problem. But that's not true. because if you only show UV, you also get hidden full responses. Yet light dots go up a little bit, and I think that's because prey capture in zebrafish is a very UV heavy behavior, but even that is less than half compared to white, right? So even prey capture isn't UV only, it's just UV helps kind of system. So what's going on? Obviously we tried more colors, more color combinations. And I just want to make a point just how strong this is. For gratings, it's extremely strong. For dots, whenever there's UV, you get some activation. But basically you get, this is white, works really well. Anything else works very badly. The best thing after white that you can do is this, which is white minus the blue. Leaving out the blue doesn't seem to hurt as much, but even that hurts, right? And it's not a lot of light. So clearly this is a white biased output to the brain, or at least the brain representation of the stimulus is white biased. Dots is completely lost. I mean, this is a few percent, and you can work with this, right? But this is not the native response. This is the native response. So what's going on? Well, this being zebrafish, you can do things that you can't do in other animals, and that is you can break things without getting into trouble. So here is a wild type fish. What do you think would happen if you were to genetically ablate the red cones acutely, so there's no developmental anything? You just take a fish, take out the red cones. What should happen? This. Okay? Almost completely lose the grating response. And actually this is, so the significance is always versus 0, not against here, right? So this is significantly more than 0. This is not significantly more than 0. The dots is still significant, and there are still a few dot responses, but hopefully you can see that this is much worse than this. Okay, so the red cone is basically most of it. The green cone is not most of it. You take out the green cone, not only are responses still there, whether they go up or down, you can debate, but actually if you just, these maps are not just the number, but the number multiplied by the amplitude of responses. And if you take that as a factor, then actually at least the light dot responses go up when you kill the green cone, right? So the green cone isn't there to drive these responses. If anything, it's there to stop them a bit.
Right, can you remind me the ratio of the different cone plus?
In the baby zebrafish, they're almost 1 to 1 to 1 to 1, not quite, but roughly there, right? So the number of cones are not crazily different. Here's what happens when you take out the blue cone. It's a bit like the green, except that now the gratings are a little bit more potentiated than the dots. But again, it's still there. This fish can still see. Take out the UV cone. It's almost like the red cone. It's just the other way around a bit. Now the gratings are a little bit alive and the dots are less alive, even though they come out as non-significant. Okay, so red and UV matters. Green and blue do weird stuff. Okay, so, but we do have some shrapnel left, both in the red and the UV case. So what happens if you take out both? Then it's completely toast. Okay, so the red and UV cones are necessary for this, certainly, and I'll show you later, it's the whole visual system. If you take out the middle two, you clean up the text loop, right? Get potentiation in both, and everything looks pretty much intact. So green and blue cones not necessary nor sufficient for vision. They must be doing something else. And actually, what's kind of cool is there's an additional phenotype that I didn't talk about. So when you play in a control fish dark dots, they don't love it. There are responses, it's not many, but on a map like this, they come out as basically nothing. If you take out this middle system, they come back, right? So the zebrafish has some sort of circuitry by which it takes its middle cones and suppresses the dark dot response. Maybe different species of fish has the light response, whatever. The point is that you take out the cones and stuff comes back. Stuff gets more, not less. Okay. So remember, I talked about the white effect in the beginning. So let's look at this at a bigger picture. So this is now many colors. We're looking at grating responses, and we've always got the white, the controls, and the green-blue double ablated. And hopefully you can appreciate that you're starting to potentiate, especially in the long wavelength range, the responses to grating. So for example, if you've got a control fish and you give it red gratings, which is sort of the standard stimulus used in the field, then you get this little hump here. And then if you take out the green-blue cone, you rescue it almost to control levels. Okay? So the presence of this green-blue system causes the white effect. Yeah, you take it out, the zebrafish vision is no longer white biased. So we're wondering, how can we map this onto the different cone types? The nice thing is that we have spectral sensitivity functions of the cones, and we can therefore take each of these stimuli, convolve them with our spectral sensitivity functions, and make predictions of how the cones would respond to the different colors. And then once we have that, we can use those cones and fit a model and basically predict which cones probably underlie which kind of things. So here is, for example, a model trying to explain in a green-blue ablated fish the grating response, which is this line. So the way to read this is, here would be red cone only, here would be UV cone only, here would be both, and these would be opponent, and these would be both negative, right? And hopefully you can see very clearly that all of the good model fits end up on this side, which is basically red only, and maybe a little bit tilted upwards, which tells us that the system is mostly red with a little bit of UV mixed in for fun. Okay? And then we can see, given that we've now looked at this, we can see, well, what's the remainder? What's this difference that we're getting here? Because that must come from these two cones. And if you do that and you fit the model, then it falls into the bottom quadrant, which is green minus blue, right? So it's not like the green and blue system are working together. They're working against each other to regulate the red UV. So it's like
the difference between this slide and the one before.
in this slide, we're fitting the green-blue ablated, right? So basically what's left after you've taken out the system.
Okay.
And that you can explain with a red cone fairly well. Whereas this you can't explain with a red cone. You need the green-blue and you need the green-blue in this sort of configuration. Green positive, but blue negative. Yeah. So it kind of suggests that we've got here, again, this system positive, and then here a regulation, and actually these two are fighting with each other for the right to regulate. So it's green minus blue system. Okay, so far so good. This is great things. What about dots? Dots is actually even clearer. So here's the dot tuning functions. If you look at the ablated version, you get a very clear red plus UV, a little bit biased towards UV, but basically both cones are important. And if you look at the remaining response in the control, it's very clearly green minus blue. So again, same architecture, gives you a slightly different tuning function because the weights are different, but the architecture is the same. Now, what about behavior? So we thought, well, the easiest behavior we can do is nothing. We just put the fish in the dish and we watch them. Let's see what they do. So here's some control fish. This is probably the most boring bit. They swim around and they do the bouts and then do a little turn and then they do some bouts. So you can count the bouts, you can count the turns, you can see how many they do. You can do this in the light, you can do it in the dark. And this is the known phenotype of, it's not phenotype, this is what fish do. When you put them in the light, they don't move so much. You put them in the dark, they move a bit more. The same. So now we take out the cones. And what I think is very cute is that we basically get a light phenotype and dark phenotype. Right? So the red cones, no matter what you do, if you show them light or if you don't show them the light, they behave like a control fish that is in the dark. So it's almost like they don't see, right? They always think it's dark. The UV cones are the other way around. If you take out the UV cone, they behave no matter what you do as if they are in the light. So it almost suggests that the red UV system has a push-pull on the behavioral decision as, do I respond as if I'm in the light or in the dark? So maybe it tells the fish, it has a sort of an underlying behavioral state type thing, something we're looking into. If you're killing the blue by itself, it's a little bit like the controls. They're all right in the light, and then they almost reach the dark state, but not quite. The greens are completely broken. They go the wrong way around. It's not significant, but it's certainly not doing the right thing. But what I think is my favorite is that if you apply both, you kind of rescue them, right? So the control, to remember, goes from here to here, and the green-blue go from here to here. So they're almost like the controls. And I think what the key thing to notice here is that the double ablated fish are better than the individual ablated fish. You take out only the green system, they're a bit shit. You take out only the blue system, they're a bit shit. You take out both, they're kind of okay. Yeah. So it's like a misregulation when you have only one out. But when you take out both, it's all right again. There's a very clear phenotype of the double ablated ones though, and it's this one. So instead of counting the number of turns, you can just see how much they turn. Like, do that turn like this, or do that turn like this? They do good control fish. And the double ablated ones, they're terrible, they do this. Yeah, it's a very striking phenotype. It's a lot of fun to watch. So they can do fishy things, but they're sort of a bit overexcitable, right? It's almost as if they're lacking the regulation to do the right thing or to do a modest thing. Okay, so that's spontaneous behavior. So what happens if you start stimulating them? Optomotor is fairly easy behavioral test, so we just adopted this setup, published from Florian Engel's lab. And basically you do an optomotor response. You watch the fish, you give it some stripes, turns, you quantify. Controls are very good at it. Red ablated fish are very bad at it. So far, so good. UV ablated fish are very bad at it. Makes sense. The green blue, they're also kind of bad, but notice that they're better than these guys. But where it becomes really interesting is you take out the green by itself, absolutely terrible. Take out the blue by itself, not so bad. And notice that both of these are significantly different from the double ablated phenotype, right? So again, it's a sort of rescue of the green effect. The blue isn't so involved, doesn't matter so much, but the green really breaks it. And if you want the green from not breaking it, you also need to take out the blue. So it's the green-blue system, they're talking to each other. And if you take out one, it goes wrong. Okay, so this is the phenotype I just talked about, where they turn too much. This is spontaneous. Well, it turns out that if you do stimulate them, it overrides it. I mean, some of the mutation phenotypes do wrong things a little bit, but the scale is a completely different one. So they're like 10% turn too much. It's just the numbers are so high that it becomes not significant, right? But hopefully you can appreciate that the basic phenotype is essentially gone, right? So these green-blue ablated fish, in the absence of a systematic stimulus, they get overexcited. Then you give them something easy to do, it's like our first stripes. And they're okay, they can do it. Yeah? Okay. So that's optomotor response. I just want to point out, so I haven't got the data here because it's literally coming right now, but this was done in a setup that doesn't do UV. This is a red, green, blue setup. It's a standard setup with a standard screen and everything. And we thought, what's up with that? And we thought, why are these so bad? They're meant to be better. So what we have done now is we have set up an aquarium outside and we've put it in the sun, which sometimes comes out in this country, as you might have witnessed. And we've put a big conveyor belt type thing under the tank and a camera above the tank and like try basically a natural type thing. And if you do that, not only are the green, blue, are these double ablated fish completely the same as these guys. These two are still terrible. But what's actually really cool is if you then increase the water depth, the controls stop responding and the green, blue ablated still respond. So basically this distance idea that I was pointing out, it looks like it actually plays out even in something as silly as an optomotor response, right? It's not just a prey capture thing or whatever, where you might expect that this is important. Like something as fundamental as optomotor, it seems to work. We just need the numbers to make a nice plot and see if it comes out as significant. Okay, so this is optomotor, but we wanted to test if it generalizes across other behaviors. So here is perhaps even more ancestral behaviors, phototaxis. Right, phototaxis is you switch on the light, you check out if the fish go towards or away from the light. And turns out that in zebrafish, it's a spectral behavior. So if you give them UV light, they go towards it. If you give them blue light, they don't care. If green light, they go towards it again, and red, they love it. Right, so that's the spectral tuning function of your controls. You take out the red cone, this time it doesn't matter. So this is one behavior where the red cone doesn't seem to be so important. But you take out the UV cone, then it breaks. So it seems to be a UV cone behavior. But again, where I think it becomes really interesting. Oh yeah, and I should point out that phototaxis is in part driven by the cones, but in part driven by melanopsin system. So that's intrinsically photosensitive ganglion cells. Yeah. But those get input from cones still. So it's still the cones are involved. But where it becomes really interesting is if you take out the green cones, they love it. They love a bit of phototaxis. Even at the wrong colors. They always go for the light. You take out the blue, they hate it. They don't want to do it at all. You take out both, they're fine. Yeah. So again, even in something as ancestral as phototaxis, which doesn't even need an eye, you don't need an eye for phototaxis. You just need to be able to measure how much light there is and start moving. Yeah. Even there, it still seems to be the case. So it looks like you've got this regulatory system in place again. So what I would then propose is that this is probably not right, at least for fish. And you've got some sort of core architecture where you've got this core system. Sometimes the red is important, sometimes the UV is important. You can recruit melanopsin circuits, but there's a core drive and there's the regulatory drive. And the regulation always comes from the green-blue system, and it seems that it's always in competition with each other, such that when you damage it half, it's bad. If you damage both, it's fine again-ish. Now, in fairly rapid closing, hopefully, let me give you some perspective on what I think this might mean for animals that are not zebrafish. So, ancestral state, 4 cones, quite clear. I think they're in composite. So this is the model. If you look at basically all of the fish, they more or less all have it. Sharks and rays are weird. And many of them live at depth and they've gone through weird bottlenecks. So if we ignore the sharks and rays, you can probably say that all the primitively aquatic lineages have the system. Then you've got this branch. This branch is everybody that's ever walked onto land that isn't a mammal. Okay, what happens here is they almost all have it, except for weird secondarily aquatic species like frogs. But what happened if the tetrapods come here? Tetrapods are things with 4 limbs. They already have lungs, they already have big eyes, stuff is changing, but they still live in the water for quite a long time. Then they start becoming amphibious. One branch of that is amphibians. And the other big branch of that is basically everything that would become reptiles, birds, that sort of thing. And they also stayed aquatic for ages. So between tetrapod and this line, we've got 100 million years of buffer window to adapt to the idea of, I don't live in the water anymore. Let's see if I can rearrange my visual circuits to use this ancestral system for something new. And they seem to have all managed to keep it. And the branch that's broken off here is the mammals. And it looks like the mammals didn't have this buffer zone. The mammals very quickly went from completely aquatic, root here, to completely terrestrial. So I would posit that it was probably related to the fact that mammals went very rapidly onto land. There wasn't time to evolve new strategies to use the system. Because it is a subtractive negative inhibitory system, it's probably bad for signals and noise. So if it doesn't give you benefits, you should get rid of it. And I would posit that these early synapsids, which eventually would become mammals, lost it. And that, just to make the point, I've got this nocturnal bottleneck in here. You might have heard of the nocturnal bottleneck theory. It's the idea that mammals lost their cones, those cones, during the age of the dinosaurs because they were nocturnal. Because the dinosaurs were diurnal, they would eat the mammals that would come out during the day, so everything was nocturnal. At night, you don't see color, but there's not enough light to see color, therefore you lose the cones. That's the ancient theory. But there's many, it's just wrong, but let me just give you 2 reasons for it. One, the actual window for cone loss, where we got any insights of, because we know the cones are present here, because all of these branches have it. We don't know, it's like if the platypus thing is wrong, then none of this branch, like it's possible they got lost here rather than here, which is what's always proposed. Okay, so the window for cone loss is very broad. But at the very latest, even if platypus has blue, the blue cone must have been lost when platypus split. Actually, it should be here. So that means that the nocturnal bottleneck doesn't align with the possible window of cone loss. So it's not very plausible there. And what was my other argument? I'm getting confused now. It'll come back to me. There's more. Anyway, I would posit that this was probably a key reason. So let me just close in making this little argument here. So you've got your zebrafish, and I think zebrafish is a fairly typical surface-dwelling fish. Of course, it's also 550 million years removed from the ancestor, but at least it still has a lifestyle that's a little bit similar to what we think our ancestors were like. We've got 4 cones and rod, and that fans out into a massive array of first-order interneurons. You've got loads of bipolar cells, something like 23 types. You've got 4 types of horizontal cells. So there's a lot, there's like a 5-fold fanning out from a network perspective. And then you've got all of these second-order neurons, and you go into your third-order neurons, which is your ganglion cells and your amacrines, and that divergence isn't all that big. By the numbers, it's about 2.5. Okay, so what then happens if you take your fish and you try to turn it into a mouse? So you just take out the system. What happens to the top? Well, the top, what happens is you get a proportional reduction. Yeah. So we've still got a ×5 fanning out. And the way it's achieved is by losing some horizontal cells and some bipolar cells. Yeah, so the numbers, the ratios are exactly the same. It's just you've lost some cones and therefore you lost some bipolar cells, some horizontal cells. But then what happens down here? Then we massively increase. Not only do mice have as many gamete cells and amacrete cells as fish, they have more. They have way more. Right? So this factor becomes 7 rather than 2.5. So I would posit that mammals lost this system very early in their evolutionary history, coming out of the water, not having a use for it. But they still needed to do the things that the system does, which is like get distance right and compute interesting things that I haven't thought of, which I'm sure that these counts will do still. So they needed to evolve compensatory strategies. And one of the ways of doing that is to put different circuits in. And what better circuit to use than an amacrete cell, which is highly diverse. It contains all the right bits in the retina to do similar kinds of computations. And it seems a very, I mean, I've used the fish versus mouse example here, but it's a mammal example. Mammals have crazy numbers down here. And if you look at something like a bird or a reptile, they still have the entire complement. They actually have grown the complement. There's new count types. This is still sort of factor 5, sometimes factor 4 up here. And then there's an intermediate down here. So the birds and the reptiles, they seem to sort of straddle two worlds of the this sort of early divergence in the fish and the late divergence in the mammal. So there's something to be thought about from a network evolution perspective here. So this is actually my in case there's time, which there isn't. So I will skip it. And I will thank everybody. I apologize for going slightly over. And yeah, thanks.
Thanks, Tom, for a great talk. Are there any questions? Yeah, Matteo.
I have two questions. One is when you were doing the calcium imaging, the gratings and the dots were moving stimuli, right?
Yep.
So drifting gratings as well. So could it be that this applies to drifting images, but not still images that the fish sees?
So there are very few. So what you might have noticed in the stimulus is that there's a static grating, then a moving grating back to static. The responses are almost all to the moving. So I think the transition from like a black screen to a stripy screen that doesn't move is, I mean, yes, the fish sees it, but it represents it with a handful of neurons. Whereas if you start moving it, then it goes. So yes, what I've said applies to moving stimuli, but I think in part of that is because non-moving stimuli are, well, they're rare. And maybe it's less difficult to represent them, so you don't need many neurons.
And the other thing I was wondering, so I always thought on your talk, I guess, that the way I estimate distance mainly is because things get more blurry. And I mean, it's interesting that you suggest fish at least might use changing color as well, right? Color appearance. Do you have any way to estimate which one decays faster, like to say that the change in how white or how colorful something appears changes faster than how they blur?
Yeah, I mean, I don't know if it's efficient to jump back to that slide, but actually, I can probably just scroll it up. I mean, if you look at my silly little movie, where is it? Oh, no, it's not about it. This one. Doesn't play in the GIF yet. So, I mean, you can see the blurriness in the brightness index, right? But I would argue that this effect is stronger. So I think the whiteness effect it works over shorter distances. Mm-hmm. I mean, this is anecdotal. I haven't quantified this, but this is my hunch.
Any other, Nicole?
So you mentioned a couple of times that you think that the monitoring finding they don't have UV cones anymore, being sort of a weird mammalian thing, maybe not being right. So do you think it's that they have the ancestral UV cone, but then turned on the SWS2 opsin and lost the SWS1?
Yeah, that's what I think. Yeah. It's just so the evidence for the platypus blue cone rather than UV cone is the presence of the gene for the opsin in the genome. That's it. Right? There's no one's looked even in the retina if it's so yeah, the evidence is weak.
Okay, any other? Yeah,
If there's time.
yeah, one last question, then let's go with that.
Okay, I don't want to be last.
No, you're good. You're good.
A question about the mouse. Because the mouse seems odd in that it's got these UV photoreceptors that are looking mostly at the sky, I think. And the greenish ones, which are the gray for you, are looking mostly at the earth or down. So what's your view with that?
Well, this is one of the things that are on my optional slides. So thank you for that.
I will take my payment later.
Okay, so here's my mouse bit. I'll try to zip through it very quickly. So this is basically what it is described. So first of all, what's happening is that the real UV cones still exist. And the red cones have a green or UV sensitive option depending on location. So there's actually two versions of the UV sensitive cone, the original red one and the original UV one. So that's the first critical distinction. Then they are positioned in different places. The top of the retina is standard. Bottom of the retina is the weird one because that has the real UV cones, the fake UV cones, and the occasional still green cone, which is red, just to confuse you. So what happens when you see the video? Oh, it's not. Okay, so this is data from Thomas Euler's lab. Basically, what they did is they built a camera that sees in mouse wavelengths, green and UV, and just took videos of the same scene. Here's the green view, here's the UV view, and basically this is what it would do, right? So you've got a green. So I've just greened it out here, right? So this is what it would see, and this is what it would see. Sort of as a sort of glued together picture. And hopefully what you can appreciate is that UV picture is better than the green picture. It's sharper, right? So above the air, UV gives beautiful silhouettes, very nice. But below below is terrible because nothing, UV light doesn't bounce. UV light silhouettes, right? So anything on the ground, you don't want to use UV for it. So it makes sense from ecological view to do that. But okay, so this is the world that the mouse faces itself, right? It's like, okay, I need to see this. How do I do it? Do I take my ancestral UV system, completely rewire how my retina work and point them at the sky in order to use this niceness? Or do I just take my red system and put a UV hat so that the UV system sees so that the red system sees in UV, but codes like a red system? That seems like a much simpler strategy to do this, yeah? And then actually, quite recently, they've discovered that the true UV cones, the ancestral ones, have a massive splotch in the bottom of the retina looking up. That's different from that distribution that you just saw. This is the real UV cones. They exist everywhere, but there's a huge hotspot. That hotspot looks at the sky, and I think it looks for birds. And here's a very nice video that, again, from Thomas Euler's paper. So they've got the green and UV, and up here you see a drone. And it's really hard to see it in the green channel, but in the UV channel, it's really easy to see, right? So if you're using your UV channel to look at the sky, not only to get the nice silhouetting from the trees and the grass and everything, but also see your predatory birds better, right? But it's nice to do it better, or you can use your mega sensitivity trick of the UV system to see it even better. I mean, it's obviously just fake enhanced, but the point is that if you take your real UV system and you really crank up the gain and look and point it at the sky, you will see your birds. So I think that's in part what it's doing. And here's some data from how they respond. So here, at the time, we couldn't distinguish the real and fake UV cones in the mouse. But basically what we found is we found some photoreceptors that are very linear and respond to green. Some photoreceptors that are very linear and respond to UV and not green. And then we found some photoreceptors that were extremely nonlinear and biased to dark contrasts. They were always UV. Right? So we at the time couldn't tell if these are the real UV cones and the other ones were the fake UV cones, but I suspect they are. So we're testing that now. But that's basically what I think is going on in France.
Mice picked up a lot of rods because they also co-opted some like UV cone based system. Have any thoughts on that or whether that's real, you think?
Can you expand on?
So Ancestral lab did this a while ago, this is a few years ago now, where they basically found that some early rod precursors started, or what they determined to be rod precursors, were trying to turn on UV options, but then they wouldn't continue into a cone fate, they would switch them to a rod fate. So this idea that they were taking cones that were destined to be a cone, making them into rods instead as part of the nocturnal bottleneck solution.
So I mean, I can't give you anything concrete about that, but I think there is it comes back to the question of where the rods and cones come from, I think. And I think if you do, we've done this, if you do transcriptomics across all the rods and all the cones and all the species you can get your hands on, and you do clever clustering, and you sort of compare what is most like the next, you'll find that the rods are by far the most molecularly distant photoreceptor. Like you've got all the cones and then you've got the rods. So that suggests that if they come from a single precursor, once upon a time, the rods are the first thing to split off. And then the cones follow spectral order. The red ones are like the green ones are like the blue ones, and the blue ones are like the UV ones. So probably either the UV ones or the red ones are the original one. If it's the UV one, it gives you a rod, and towards the other cones, that might explain it. I do have reasons to suspect, though, that the red one is the original one. So I think there's something to be.
Cool. Great. Thank you very much, Tom, again. And after stop. Okay. Thank you very much. Notice if there's any more questions.